old images, that there are categories of microtubule polymer that are very rich or very deficient in these modified forms. Now these are the tyrosinated and detyrosinated domains. Acetylation looks a little bit different. The acetylated tubulin seems to appear in the form of these patches along the length of microtubules. So nowadays there are some tools for manipulating at least acetylation pretty effectively. So HDAC6 is a deacetylase, and we can overexpress HDAC6. And then tubicin is a drug that inhibits HDAC6. So if you overexpress HDAC6, you're going to get a lot less acetylation. So this is an immunostatin for acetylated tubulin. This guy right here that's overexpressing HDAC6, notice that it contains almost no acetylated tubulin state. This one contains a little bit. Then if you treat with tubicin to inhibit HDAC6, then you do your acetylated tubulin staining. They're screaming bright. So we have the experimental ability to increase acetylation or decrease it. And to make a long story short, that if we elevate microtubule acetylation, then we get more souring by ketamine. If we lower microtubule acetylation, we get less souring. And if we look very specifically at where the severing occurs, this is ketamine, this is spastin, and we look very specifically at where the severing occurs, notice with ketamine, this is a acetylated tubulin. I hope you can see this. It's a little hard to see. But that the severing occurs where there's a acetylated tubulin. Whereas the spastin, this seems more random. So here's a cup, and in this region you don't really see the acetylated tubulin staining. So what the conclusion here is that ketamine really seems quite specific for acetylated tubulin, microtubules that are rich in acetylated tubulin, where spastin doesn't really seem to care so much. So we take these studies in neurons. Yes?
So that would mean that ketamine would lead to more stabilized branching, because otherwise one would be more stable in forming the branches.
Yeah, that's a really good point. Because what is the purpose of microtubule severing? And at least with regard to the cut and run model, the purpose is to create short, mobile microtubules that can move into a new branch, that can move out from the centrosome, that can live in order to carry out a purpose. So if you imagine breaking up a highly dynamic microtubule, it's just going to disassemble. So there's not much reason for doing so. So the idea is, if you can target the microtubule severing to more stable microtubules, then yes, those short pieces will persist. They will live in order to be able to transport into new branches, into axons or dendrites. So having this mechanism for preferring stable microtubules through a preference for acetylated tubulin makes good sense for that reason.
And it could be like ketamine gives rise to acetylated tubules as such, and they could act as nucleating center using the tubulin monomers generated by spasm.
Yes, exactly. Yeah. That by ketamine having a preference to break up acetylated microtubules, the end result will be a high number of short fragments that are stable enough to resist depolymerization and live another day to act as nucleation sites. Is that? Yeah.
Does tau have a different affinities for acetylated tubulin, or does tau has a different physics affinities for acetylated and non-acetylated tubulin?
I don't know. it's a good question. Does tau have a different affinity for acetylated or deacetylated tubulin? I don't think there's any evidence on that I know about. So moving on to neurons, we did our 2 experiments. Remember, with HDAC6, we can substantially lower the content of acetylated tubulin. Then with tubicin, we can significantly increase. This is 24 hours of ketonic overexpression. Now look here. This is HDAC6. We've overexpressed ketonic for 24 hours. And notice the microtubule stain is still very robust. The microtubules are really not being acetylated very much because they're not acetylated. By contrast, you can barely see the stain. In the tubicin case, because in the tubicin case, you have made the microtubules very, very acetylated. Way. You made the microtubule. I think I got this backwards. So in the HDAC6 case, you made the microtubules very, very unacetylated. So yeah. Okay. So the difference is you've got tau present. The difference is that with the tau present, for axons, it doesn't care so much. It's only the dendrites that it cares. So it's a little bit of a complicated situation. So that makes the story a bit complicated. But for axons, notice that all these bars are similar because whether you have the control, the HDAC6, the tubicin, the ketone, whatever you do, it's very similar because they're tau rich. Whereas dendrites really follow the same rules as the fibroblasts. So the control, the microtubule levels are high, the HDAC6, the levels remain high. Tubicin, now with tubicin, you made the microtubules very acetylated. Now here, with ketone, you lowered the levels. With the ketone plus the HDAC6, now the levels do not diminish because the microtubules are very unacetylated. But with the tubicin, now you get a decrease. Now you get a decrease. If you overexpress tau, such that tau goes into axons and dendrites, then it's very protective. It doesn't matter what you do. So the neurons are a rather unusual situation because they have to pay attention to 2 different factors. They have to pay attention to acetylation status and the presence or absence of tau on the microtubules. So various levels of regulation. One is the presence of various separate proteins, about 7 in total, with somewhat different properties. Another is the levels of distribution of these separate proteins. Some are widely distributed, others may be concentrated at branch points, for example. The sensitivity to tau and other sorts of factors. And again, this is not been done exhaustively. Spasm, for example, may be very sensitive to the presence of, say, for example, double cord microtubules. So this is another level of regulation that really needs a lot more work. And then acetylation, other tubular modifications may be a factor. Karsten Jahnke has done some really excellent work showing that spasm is highly sensitive to the levels of polymetabolated tubular microtubules, for example. And then another possibility is regulated degradation of these proteins. Now, what about disease? And I want to talk a bit about hereditary spastic paraplegia and then a bit about tauopathy. And I'm going to ponder, is hereditary spastic paraplegia not enough severing? Is that really at the heart of it? And is tauopathy really too much severing? So, getting back to hereditary spastic paraplegia, This is an autosomal dominant disease, mutations in the spastin gene, very specifically causes degeneration of cortical spinal tracts. There are no developmental problems, no problems in the peripheral nervous system. So it seems highly specific to the central nervous system. It's adult onset, caused by mutations to spastin. Now, here we have a mystery. Why is it that this is adult onset? Why is it only the cortical spinal tracts? Now, the community studying hereditary spastic paraplegia mainly are people who come at the disease not from a microtubule background. And this is really a good lesson about how having a background in basic science and coming at these issues with a large body of basic science information can really be helpful toward having a solid insight into the etiology of the disease. So, the theory that most people in the community really latched onto was a loss of function scenario, a haploinsufficiency. So how many know what haploinsufficiency is? So what haploinsufficiency, can you explain it? What's haploinsufficiency?
That one dosage of the same gene is not sufficient.
Exactly. So you've got two spastin genes, and the idea is one of them is mutated such that the protein created by that gene, that mutated protein, doesn't work. Then you're at the mercy of roughly half the amount of total protein. And that's not enough. That's not enough to keep you healthy. And if you look at the hereditary spastic paraplegia literature, ever since spastin was discovered to be a severin protein, people have latched onto this haploinsufficiency idea. In truth, not all that many diseases are due to haploinsufficiency. It's actually kind of rare, particularly for enzymes, because usually enzymes are made in excess so that you presumably have enough of it, even if you only have half. But this becomes even more mysterious for spastin, because spastin would be presumably especially important for development, right? There are a lot of things that have to happen during development. Branch formation, and growth cone motility, release from the centrosome of microtubules, a lot of different developmental events. And yet, developmentally, there are no symptoms. The patients are perfectly okay. Moreover, why would only the cortical spinal tracts be sensitive? Why not, say, the sciatic nerve, which is a peripheral nerve, but is quite long? I mean, some people make the argument that, the reason why it's the cortical spinal tracts is they're really quite long. They're long motor axons that go to the limbs. Well, there are some long peripheral nerve axons as well. So my feeling is that once people in the community who did not really have a basic science background, once they saw, it's a microtubule severin protein, there must be something wrong with the microtubules. There must not be enough microtubule severin. So what we did is we looked at a few parameters in this. One was we looked at the absolute levels. So this isn't just proportions, it's absolute levels. We took our antibodies and we calibrated them against the recombinant protein. And during development, there's cationic, there's spastin. And the levels during development of cationic progressively go down. the levels of spastin are pretty steady. The levels of spastin are not going down appreciably. So one argument you could make is that all the way during development, maybe ketamine can compensate for spastin, but then once you reach the adult years, then maybe not. Maybe there's not enough spastin and ketamine in combination to induce sufficient severing to keep the axon healthy. So maybe. But what I'd like to propose to you is a gain of function mechanism. Now, let me tell you, if there's any person anywhere in the world who should be emotionally invested in a loss of function mechanism for hereditary spastic paraplegia, it should be me. Because I put so much of my career into the importance of microtubule severing in the nervous system. And yet, I just don't think it makes a lot of sense for the reasons I just explained. Let me tell you why I think a gain of function makes sense. What do I mean by gain of function? Can anyone know what gain of function means?
Is it functionally more active?
Not really. Acquires a new function. So when the spastin mutant, when the spastin protein has the mutation, it acquires a new function. That sounds like a good thing, but no. When I say it acquires a new function, I mean a bad thing. I mean it's doing something it's not supposed to do. So loss of function diseases are when there's not enough of the protein to carry out what it's supposed to do. A gain of function disease is when that mutant protein is doing things that are bad, that it's not supposed to do. And the key to all of this, the mystery, I think, is all about these two isoforms. So remember the shorter isoform that comes from encoding by the second coder, almost all the spastin that you've ever found in your body, and it's not just in neurons, it's throughout the body. It's in skin cells, it's in all your cells. The vast majority of that spastin is the shorter isoform, M85 in rodents or M87 in humans. This longer isoform, when we Western blot, this is cerebral cortex, this is spinal cord. This is E18, P0 is embryonic, 18, that's newborn, that's P12, this is mouse. Spinal cord. Here's M85. M1 would appear up here. You just don't see it. You just don't see it. Now look at spinal cord. Spinal cord is where the cortical spinal tracts lie. E18, nothing. P0, nothing. P12, nothing. Look at that. You see that? In the adult, you start to see M1. Adult spinal cord, adult cortical spinal tracts. My theory is that when M85 bears the mutation, it may not work. It may not sever microtubules, but it's not causing any problem. When M1 bears the mutation, now you've got a cytotoxic protein. Now it can cause problems. Because that is present in the right place at the right time. Adult spinal cord. And here you see it even better. That's even better. You see M1. Let's take a break for 10 minutes and then I'll finish up.
Yeah, please
Take a break for 10 minutes. T-brain that a loss of function scenario doesn't really make a whole lot of sense. But a gain of function scenario might make sense. So my hypothesis is that mutations of spasm might just be particularly dangerous if they occur with M1. And the difference between M1 and M85 is a region that is really very hydrophobic and a region that might be very prone to misfolding. And as you remember from previous talks from Dr. Brady and others, that when proteins misfold, then they can start imposing some real toxic effects, at least potentially. So what we've done here is we've made some mutant forms, and these are just stock forms. We've just simply taken the M85, the M1, and we've made what amounts to a truncation mutation, because a whole variety, it's actually about 200 or more different mutations of spastin that give rise to the hereditary spastic paraplegia. Some are full-length mutations where the patient has the entire spastic molecule, which is a point mutation. Then others are called truncating mutations that result in shorter proteins. So we created some of these shorter proteins, and we did a couple of experiments. And this experiment that you see in the lower panel was done with Gerardo Morfini and Scott Brady. Very similar to the squid axoplasm studies that Scott showed a few days ago. And what you can appreciate from this is that with the M85, really no effect. So we infuse the squid axoplasm with the mutant form of M85, and both the forward and the backward transport of vesicles appears pretty normal. But here, notice the downward slope in both the anterograde and the retrograde transport. So, this mutant form of spastin, when infused with axoplasm, seems to be creating a deficit in fast axonal transport. So, for haploinsufficiency people, this is a very strange sort of result. But gain of function makes good sense, very similar to what Scott Brady reported, say, with mutant with tau fibrils, with other mutant proteins, he studied with Huntington. Here, we did a cell biological experiment where we took control neurons. Here, you see we've expressed the M85 mutant. Looks pretty good. Long axon, lots of little galpodia, minor processes, not much difference in axon length. But look when we express the M1 stop. The cell body is smaller, the axon is shorter, and it just doesn't look good at all. Now, this was a mutant mouse M85. We reported this a couple years ago. More recently, in my lab, what we've done is we've generated a full variety of mutations using M87, specifically for human. When we've done this, we've made mutations. We've made the exact mutation. We haven't just created this artificial stop protein, but we've put the exact mutations in a whole variety of different patients in these constructs. And there are even a few mutations that are actually not function blocking. They're actually not function blocking, but they still cause the disease. And that within itself should obliterate the haploinsufficiency, the loss of function model. Because those mutations do function properly. They're not in the ATP region, they're in some other region of this spastin molecule. Yet they still cause the disease. And when we work with the human, and we introduce these exact mutations, these mutant spastins are far more toxic than you even see here. We can barely express them and they kill cells. We express them in Drosophila, and the flies just look terrible. You've got to use specific promoters that induce the expression only the adult fly, only in certain neurons, or you don't even have a fly. So I think what this shows is that spastin is a molecule, at least M1 spastin is a molecule, that if it's misfolded, it's tragically toxic. Which is probably why, evolutionarily, spastin developed that second start codon. Is so that in the vast majority of cases, the cells could dodge, even playing fire with M1, because M1 spastin is a protein that is so prone to misfolding. Now that begs the question that I'm sure many of you are thinking, why is it that adult corticospinal neurons will start expressing M1 spastin? Why do they make this trouble for themselves? I don't know. I don't know. Does it have some kind of a function that evolution needed to hold on to? That the corticospinal neurons need to have some of this M1 spastin? Or in some way, was it just a mistake, just a tragic mistake that was made along the way, that the human body would be far better off if it had never preserved that initial start codon? We don't know the answer to that. But I think, at least to my mind, that these data are really compelling. The haploinsufficiency not only is illogical for this particular disease, but that the weight of evidence is really against it. There's also a spastin knockout mouse that seems perfectly fine. Knockout spastin in individual neurons in culture, they do okay. Maybe not quite as many branches, but they seem to do okay. All the other neurons of the body seem fine in patients with hereditary spastic paraplegia. So the idea that there's something somehow awry in the nervous system, when it's also expressing 6 other cellular proteins, I think stretches credulity. And I think this gain of function model not only fits logic and fits the evidence better, it also puts hereditary spastic paraplegia more squarely in the category of other neurodegenerative diseases, such as Alzheimer's disease, such as ALS, Parkinson's, Huntington's, other diseases that are caused by misfolded proteins. And I bring up this story about haploinsufficiency not to disparage the investigators who favor this model, but I think really to point out the incredible value of basic science. Now, we all contribute at different levels, and clinician scientists who are focused mainly on disease, they have a great deal to offer as well. But having this body of basic science knowledge is so incredibly important, so useful, that when I think back to, remember I said 1993, when I was thinking to myself, how do branches form? How do microtubules get to a branch without calling back to the centrosome? How do they do that? And I thought, well, maybe it's because microtubules are broken up into little pieces. Nobody had ever discovered even katana yet at the time. But it made good sense. And at the time that I thought about that, at the time that we did all those tedious serial reconstructions, electron micrographs, thin sectioning, put all those back together again to show that indeed microtubules are separate at branch points. Was I thinking that someday that would help us to understand hereditary spastic paraplegia? The thought never occurred to me. Never occurred to me. It was really the pursuit of basic science questions, the curiosity, and the wonderful curiosity that I see in all of you, and all the questions that you keep asking us. And so often we say, I don't know the answer. It's not been done yet. It's that curiosity that's going to lead to treatments and cures for all sorts of diseases and maladies and injuries that afflict the human population. So I think that's the broad lesson of this sort of thing. Now, what about too many minutes? Finish up real quick. Okay, 5 minutes. So, the idea about tauopathy, I'm going to make this really quick. This is what you see all the time. Tau comes off the microtubules, they become destabilized and they break up. Well, if the microtubules break up in Alzheimer's disease and other tauopathies, it would be terribly tragic. And they do break up. I mean, there's a loss of microtubule mass. But I'm here to tell you, as I've argued, and as Scott Brady has argued, there is really no evidence that microtubules truly destabilize when they lose their tau. There's just nothing. I mean, people have knocked out tau, microtubules don't become destabilized. Neurons contain double cortin, MAP2, MAP1B, MAP1A, stop. They contain a whole array of stabilizing proteins. So here's my idea. My idea is directly launched, directly based on the tau information that I gave you earlier, that if you deplete tau, the microtubules become very sensitive to katana. So my theory is that when the microtubules lose their tau, they're becoming progressively more sensitive to these several proteins. So they're falling apart, yeah, but not by being destabilized, by being over-severed. I'm not going to show you all these data, but that's the idea. That's the idea. So here, notice, you've got katana overexpression, you've knocked out tau, and you really lost the microtubules. So one other thing I'll show you really quickly is we heard from Jim some beautiful data on axon regeneration. Is it conceivable that we can exploit microtubule severing to augment axon regeneration? And what we found is that if we knock out fidgetin, we get a lot more axon growth. A lot more axon growth. These are adult DRGs, so this is a regeneration system. Fidgetin is another microtubule severing protein. And we get more crossing onto inhibitory substrates. Here's what I think is going on. This is just a hypothesis, and you are the first people to hear it. It's based on a little bit of data you see here. But I think what happens when you knock down fidgetin is you get less acetylated tubulin, and a little uptick in total tubulin. I think what's going on is that just as katanin targets acetylated microtubules, fidgetin targets unacetylated microtubules. Now, why should it do that? How would that work? And my idea is, you've got an axon that's full of microtubules, acetylated microtubules, deacetylated microtubules. Imagine this. The fidgetin is constantly severing the unacetylated microtubules. And that's what enables neurons, as they develop, to accumulate acetylated microtubules, because you're constantly pruning back on the unacetylated microtubules. And that's important for reaching the adult state of having a predominantly acetylated microtubule array. Think of it like a bonsai tree. You know what a bonsai tree is? You're constantly trimming it. By constantly trimming it, very specifically, you are shaping it. And here in neurons, there's katanin. Katanin like 1, katanin like 2, fidgetin, spastic, fidgetin like 1, fidgetin like 2. And they're all working a little differently. Imagine different scissors trimming away at different categories of microtubules. Tau-rich microtubules, tau-deficient microtubules, acetylated, polyglutamylated, unacetylated. And that's how you can craft the microtubule array to be what it needs to be in an axon, in a dendrite, in a developing axon, and in an adult axon. What you want to do with the adult axon, when it's injured, is restore it to a more developmental state so it grows. And that's why I think inhibiting fidgetin works to augment regeneration. All that is just a theory. Everything I just said, and I'm not going to go through these conclusions, all that is just a theory. I like it, but it's going to require a lot of work to do. So the field of microtubule separated proteins is wide open. These are really, really, really interesting proteins. And if you look at PubMed, you go to fidgetin or fidgetin-like 2, maybe 2 papers published. Almost nothing. You try to get antibodies, they're not available. So these are wide open. A lot's known about tau, a lot's known about kinesin 1. Almost nothing is known about fidgetin-like 2. There was a paper in Nature that was published a couple months ago on autism, in which the most commonly mutated genes in autism were categorized. Number 3 on the list was katanin-like 2. Huge list, hundreds of genes. The third most common mutation. Now that's a correlation, it's not a cause. But in autism, there are flaws in neuronal migration. And I'm thinking, what if katanin-like 2 has a really important role in neuronal migration? So let's start studying at neuronal migration. So I think this goes to illustrate that the basic science behind these proteins, behind many proteins, broadly open. And if you choose to take up the call and to study some of these, the rewards will not only be opening the door to basic science, but truly, that is, in my way of thinking, that really is the only way to one day solve the diseases and to develop strategies for treating the injuries that affect the human population. So that said, these are the people in my lab who conducted this work, and thank you for allowing me to go over time. I apologize.
I'm sorry, I have to cut down the questions now because otherwise we will all miss lunch. So we'll go straight to Peter Holland after this, and we'll come back and have your questions post lunch when Peter comes back. Thanks, Peter.